Спасибо. Так, my dears, start coming. Guys, we are about to start our lecture. Guys, who will be the first? Guys, lecture is started already. My dears, today topic of today's lecture is Rickettsiae. Oh, now you can see that people start coming to take a look at the lecture. And Mantan, you are the first one. Good afternoon, Mantan. So, Mantan, please, if you get the link to the lecture, I have a request. Please send this lecture, the link to this lecture, through VK to the students of first division and second division, if it is possible, please. Because they also need to listen this lecture. Today I read lecture for both divisions. So that's why, Mantan, I would like to ask you to send a link there, okay? I also see that Naeem is connecting from Bangladesh, is it true? And Naeem Janatun, right? So I ask you to develop a special like group link, and please send me this link because we need to do something with you regarding microbiology and immunology classes. I would, oh no, not just microbiology. So I am waiting for your link there, okay? Because I want to give you some task. So, yeah, I can see, Mantan, that it is done already. Guys, I will start lecture right now. And the thing is that today I read lecture about bacteria that we, as in the previous lecture, we will not have class for these bacteria, but questions about these bacteria will be placed in the exam questions. So another thing why we try to study this bacteria, because these bacteria are really very unusual. From the very beginning, I want you to understand that this bacteria is somewhere in between bacteria and viruses, because these bacteria are obligate intracellular parasites. Do you know what it means? So it means this is a property of the virus, but also Rickettsiae. They are not able to develop themselves outside the human or animal cell, or sometimes vector cell. So it means they cannot replicate or multiply on their own, while in the many other features they still recognized as bacteria, and they are bacteria. Understood? So, but that's very important. And the intracellular location of these microbes are seen here, all these Rickettsiae is seen here. So you see the cell full of Rickettsiae. And also I would like to draw your attention at very nice photograph, false colored scanning electron microphotograph, because it is connected with the most important property of Rickettsiae, to be able to invade the host tissues and spread in the human cell. Can you see here that Rickettsiae may cause actin-myosin rearrangement in cytoskeleton of the cell, and from the expanse of the cytoskeleton, the green one is Rickettsiae. It may get so-called tail out of the actin-myosin, and this tail will propeller, used as a propeller or tail, may give motility to Rickettsiae to spread within the cell and to outside the cell. See, that's how it works. And by the way, this is for Rickettsiae and this is for Ehrlichia. This is one of the genus that also recognize Rickettsiae-like bacteria, but not Rickettsiae itself. Okay? So now we start, and at the end of our lecture, I would like also to give some information about the prions. So, proteinaceous infectious particle. So, they are not viruses and they are not bacteria, they are just proteins. So, if you have enough time, then we also take a look at that pathogen. But now, our main task is Rickettsiae. And of course, I would like to tell a few words about those researchers who found Rickettsiae. And I would like to draw your attention to this person. This is a Howard Ricketts. He was an American pathologist who first discovered the rickettsial disease. And that human rickettsial disease was Rocky Mountain spotted fever. And that usually happens in microbiology, that disease is found first, and then the causative agent of the disease is being recovered from the patient. That happens like this. And Ricketts worked in America with the Rickettsiae that had been spread by cattle, ticks, and ticks, when they bite humans, they transmit the microbe that cause the specific spotted fever. And Howard Ricketts described it as a blood-borne microbe that undergoes bipolar staining, bipolar bacilli, and that was later named by his name. Why? Because in 1910 he showed that the disease, that time it was really very common, and he showed that epidemic typhus, also caused by similar with Rocky Mountain spotted fever causative agent, with similar organisms, and these organisms is carried not by tick but by lice. But then, in the same year, when he did this research work, so he was stricken by the typhus and died at age 39, my dears. Can you imagine? 39. Then I would like to also present you one more microbiologist, Stanislaus von Provazek, or Provacek in Russian, Provazek in Poland, in Polish. He was also a Czech parasitologist, not Polish, Czech parasitologist, and he, together with one more pathologist that was called Rohalima name was Rohalima. They discovered the pathogen of epidemic typhus and recovered it. And they had studied epidemic typhus in Serbia and Istanbul in 1914, and then later on, Provazek and Rohalima were working in German prison hospital. They both become infected with the epidemic typhus, but then Rohalima recovered and Provazek died soon after the 17th of February 1915. So then, because Rohalima, and this is Henrik da Rohalima, he is a Portuguese pathologist and microbiologist, and Rohalima became internationally famous for discovering exanthematic typhus, which is transmitted by body lice, and afflicted soldiers and prisoners of war in Europe. The infectious agent was called, and he gave this name to it, was called Rickettsiae Provazeki, as a tribute to researchers who started studying this group of microorganisms, Howard Taylor Ricketts and Stanislas von Provazek, which died who died of the disease while trying to study. With the discovery, Rohalima opened a new chapter in microbiology book. There were no Rickettsiae yet in the microbiology book of that time. Now we know that this is a very large group of very serious diseases that are called Rickettsian diseases. So, these are two distinct organisms, Rickettsiae rickettsii and Rickettsiae provazeki, discovered by Ricketts and Provazek. And they were the first of what we later shown to be an unusual genus of virus-like bacteria named Rickettsiae. And you can see the Giemsa stain of the cell infected with Rickettsiae here. So, in fact, the classification of Rickettsiae, of these types of bacteria, had been changed many times. So, because first they are very small and they are intracellular parasites, it was really difficult to grow them in the laboratory. Second, they used to cause very serious diseases. So the researchers didn't have enough time or means to culture them properly and to learn to study them properly. So when we introduce DNA analysis very much in the process of bacteria classification and studying the relationships between bacteria. So then the DNA analysis revealed that this classification initial, where Rickettsiae, Orientia, Ehrlichia, and Coxiella had been placed in one single family, Rickettsiae. But then DNA assays showed that the similarity in the genome is not that high enough to consider them as members of one single family. So several times in the past years, classification of Rickettsiae had been reconsidered. So, but irrespectively, we will take a look at new classification, but irrespectively of the family they are placed into or they belong to, those four genera quite often are discussed as distinct, taxonomically unrelated genera but also quite often we call them Rickettsiae-like organisms, and diseases are called Rickettsiae type infections. But then, in fact, they are placed now in different even subfamilies. So here you can see nowadays taxonomy of Rickettsiae. And these are the taxonomical levels, and these, how do we call the diseases, right? So in class alpha protobacteria, we have several alpha 1 class and alpha 2 class protobacteria. In alpha 1 class, we have family Rickettsiae, and the genera Rickettsiae, Orientia, and Ehrlichia are considered now as the genera of Rickettsiae family. And in alpha 2 protobacteria group, we have family Bartonellaceae, and the genus Bartonella is placed there, and the disease is called Bartonellosis. Disease is caused by Ehrlichia, called Ehrlichiosis. Disease is caused by Rickettsiae and Orientia, called the Rickettsioses. They are most closely related intracellular bacteria. So, and then we also have class gamma protobacteria, and there the genus Coxiella belongs to, and the diseases caused by members of this genus are called Coxiellosis. So mainly we will be talking about Rickettsiae, but also to some extent about Ehrlichia and Coxiella, less to Bartonella, because these different types of genera may act differently inside the host cell. So now, what are the general properties that are almost same in all those 4 genera? And because of that, they had been recognized before as members of one family. So, but we believe that this is a quite diverse group of the organisms. And if we are thinking about the origin of Rickettsiae, there's 2 theories. First theory, that's degraded, that this is a degraded bacteria that get used to the intracellular parasitism. But the second and quite sustainable theory, that is the organism that may originate from the viruses. Both theories, so I mean, they have their own plus and minus evidences, so but we won't discuss it. That for us is important. So they are all obligate intracellular parasites. And moreover, they require an arthropod vector as a part of their natural cycle. What does it mean? It means that usually they cannot directly be transmitted from animal to human, even through the vector bite. So these bacteria should propagate, should multiply in the vector. They should spend a part of their life cycle in a vector, so they do reproduce themselves in the vector tick or lice cells. So, and only then they start to be infectious and may infect the next victim. So, and other properties are they are all gram-negative bacteria, and regarding the shape, they are cocobacilli. It means they are spherical or lancet-shaped cocci, but we call them cocobacilli. Bacilli that we call cocobacilli. And they are usually strictly or obligate aerobic bacteria. Very few of them may be facultative and aerobic, but because they develop themselves in the cell, so they are aerobic bacteria. And also require for transmission the arthropod vector. So to compare the properties of Rickettsiae bacteria and viruses, so what they get from bacteria and what they get from the viruses. So the properties that make them related to bacteria, these are that they both contain DNA chromosome and may use RNA molecules for some special functions like tRNA and messenger RNA. Then they also may keep some enzymes that help them to perform fermentation. The enzyme that participate, for example, in Krebs cycle. And they also have the ribosomes for their protein synthesis. So it means they may do some functions, normal functions of the cell by themselves. So, and they also may multiply by binary fission like any other bacteria. And why we think they are bacteria? So they are sensitive to antibiotics, while viruses are not sensitive for antibiotics. So all these to the left properties given to the left, so make us thinking that they are bacteria. So because those are not typical for viruses. So while still they can keep some properties that make them related to the virus also. And the first one, of course, they are obligate intracellular parasites. They may grow in eukaryotic cells, and so in the eukaryotic cells we may see some so-called inclusion bodies or some cytopathological effects caused by these bacteria. So they are very small, and the size range from 0.3 to 2 micrometers. They are smaller than other types of bacteria. And they stain very poor with the Gram stain. So now, what is the structure and physiology of Rickettsiae? All four genera that we mentioned at the beginning, it is interestingly that they have the cell wall, and the cell wall is almost typical for gram-negative bacteria. What does it mean? It means they have peptidoglycan layer, but this layer is very thin in some of Rickettsiae and Rickettsiae-like bacteria. So, for example, and apart from this, they have in the outer membrane, they have lipopolysaccharide. So, but the toxic potential of this lipopolysaccharide is much weaker to compare with traditional gram-negative bacteria. So it means some of them, for example, Ehrlichia, may have the lipopolysaccharide don't really demonstrate any endotoxic activity, so they will not be able to cause endotoxic shock. But some, like Rickettsiae, they can cause some endotoxic activity. So then external to the outer membrane, cell wall membrane of this type of bacteria, we may see very thin polysaccharide slime layer. It is not really very heavy, thick, very well developed and shaped capsule, no. But then still, this is a layer that make them similar with bacteria. And also they may have fimbriaopili that are important in the process of genetic exchange by conjugation. So it means this bacteria may share antigenic determinants and some other genes within the population. And they don't have flagella, so they are non-motile. Intracellular location of these bacteria may vary. Of course, the most important targets are the endothelial cells. But then in the endothelial cells, they may occur in different compartments. For example, Rickettsiae and Rickettsiae are usually found free in the cytoplasm. And Ehrlichia and Coxiella usually multiply in the cytoplasmic vacuoles. What is the difference? So when they reside in cytoplasm, they are not covered, they occur directly in cytoplasm, and they are not they don't have the endoplasmic vesicle around, so endoplasmic membrane won't really will not really make the border for their replication, right? While the Ehrlichia and Coxiella, you can see that usually they are located not inside the cytoplasm, but in cytoplasmic vacuoles. And the thing is that why they would prefer to live inside the cell. Mostly Rickettsiae and Ehrlichia are unstable outside the cell, so they are very sensitive to the environmental factors. So they will die very quickly outside the human cell. While the Coxiella is a bit different. So it is highly resistant to some factors of the environment, for example, desiccation or drying, and it may remain viable in the environment for a very long while, from months to years. So that is a difference between them. So because they are all obligate intracellular parasites, it is really difficult to put them in culture and to multiply them. But that is for several reasons. First, they are very fastidious, so it means we have to develop a special models for their growth. And second, they are really very dangerous. So they cause lethal diseases, so that's why the multiplication or culture of Rickettsiae and Rickettsiae-like organisms in the laboratory is really very tricky. So, and the only reference labs assigned for, according to the United States classification, assigned for 4th and 3rd levels of biosafety are allowed to perform this growth. According to Russian Federation classification, the highest risk is level 1 and 2. So that's why culture can be used, but in very special reference labs. So they can be used, this culture can be used for diagnostic identification. How can we culture them? And that's make them more similar with viruses. And because they are obligate intracellular parasites, we have basically 3 ways of their growth. First, they may, the animal model or the animals can be infected, and the animal model, for example, mouse or some other animals, and also we call it living host or in vivo growth, that is in embryonated egg yolk sac. Usually we inoculate them into the yolk cell. That's the best place for them to multiply. And also we may perform in vitro growth when bacteria are cultured in tissue cultures or in very special cells. And you know that several cell lines can be used for their growth. For example, Vero cells, you know it very well, and some other. The examples are given for those cells that may be used for propagation of Rickettsiae. But usually, we have discussed how do we classify them, but quite often, Rickettsiae can be classified because we have so many species of Rickettsiae in the genus Rickettsiae. So then the classification, interspecies classification is quite tricky. So that's why very commonly we may group Rickettsiae according to the type of disease they cause in humans or animals. And also we consider the vector that is used in the transmission of this particular type of Rickettsiae. And you can see here that we may develop several groups. The first one is typhus group. And there we have two, Rickettsiae provazeki that causes so-called epidemic typhus, and Rickettsiae typhi that causes endemic typhus. My dear, see, we have two types of typhus, but we have also many animal bacteria that may cause diseases in humans. The Rickettsiae typhi is the one that reside in rodents. So you can see here the vertebrate hosts for these bacteria. For Rickettsiae provazeki, these are humans only because the vector of the disease is a louse. And louse feces usually serve as the infected substance that usually rubbed against the body, and if you have the bite inside, so if feces are rubbed against the bite inside, so Rickettsiae may enter through this injury in the skin. So, and because lice are only human parasites, so this disease is strictly epidemic typhus is strictly anthropootic, so it can be transmitted from human to human only, because louse lives on humans only, doesn't bind animals. So while the Rickettsiae typhi that causes endemic typhus, so it resides in rodents as a natural host for this bacteria, and it can be transmitted in rodents through flea bite and through flea feces, but also it can be transmitted by flea feces from rodents to animals, when sometimes flea may bite humans also. So I would like to draw your attention at the names epidemic and endemic typhus. So it means that epidemic typhus, Rickettsiae provazeki, may cause like very rapid spreading among humans. So it can cause a sort of epidemic. So while the Rickettsiae typhi causes really very severe disease, but it cannot spread that fast because it depends on flea bite. So, and it is usually not transmitted from human to human. So this is a zoonotic disease. Then we may see here that the so-called spotted fever group of Rickettsiae also is categorized, right? So why spotted fever? Because one of the major symptoms is rash, and the second one is very high temperature. So typhus may happen as a case with very high temperature, sometimes without the rash, while the rash also can be a very important symptom of this disease. In spotted fever group, we have several Rickettsiae. The first one, of course, is the one discovered by Ricketts. And this is Rocky Mountain spotted fever group, discovered in America, in particular geographic area, right? It is transmitted by bite of ticks. And the natural hosts for this bacteria are rodents. So this is a zoonotic infection. So then Rickettsiae acari, it causes rickettsial pox, so-called. It is called pox because the vesicles formed on the surface of the suspected cases looks like the chickenpox or smallpox vesicles at the very beginning of their development. So this disease is transmitted by bite of mouse mite. This is a special type of ticks. And the house mice is the typical vertebrate host, but then it also can be transmitted to humans. And Rickettsiae connori, that also transmitted by tick bite, and it has some potential hosts, except for rodents. The dogs are question mark. We think that it may reside, so the dogs may also be infected with the Rickettsiae connori, and it causes so-called botoneus fever. So then we take a look at the diseases called by other three Rickettsiae-like microorganisms. So these are independent genera, Rickettsiae, Ehrlichia, and Coxiella. So we have scrub typhus group. And before, the causative agent of the disease had been called Rickettsiae tsutsugamushi, that causes scrub typhus. But then it was now categorized as Rickettsiae. So now it is placed in a separate genus. And the natural hosts for these bacteria are rodents, especially rats are susceptible. And the vector that mediates the transmission from animals to humans is bite of the particular stage in the mite development of larva of mites, and it is called chigger because of the sound it may produce. So then we have a group of Ehrlichia that cause ehrlichiosis, right? So first one is Chaffensis. That is the disease caused by this bacteria. It's called human monocytic ehrlichiosis, and it transmitted by tick bite from dogs, rodents, horses sometimes, or humans from this source to new humans, right? So then we may have some more Ehrlichia and also Rickettsiae and also Anaplasma. They all, we believe, cause ehrlichiosis. And now finally we have one more group, the just ehrlichiosis caused by Ehrlichia chaffensis is more common, the rest are less common, of course. And also Coxiella burnetii that causes a disease that is called Q fever. Q because of the question mark. So as usually first disease had been discovered, and for a very long while the causative agent was unknown, stayed unknown. So but then when they identify it, they call it Coxiella burnetii, but the disease is called Q fever. It can be that the only one out of all Rickettsiae and Rickettsiae-like organisms that may spread through aerosol. So, of the animal products or tick feces. And the normal vertebrate hosts for it are wild or domestic ungulates. So, now we are going to discuss the pathogenesis of the Rickettsiae and Rickettsiae-like organisms infection. So Rickettsiae species are transmitted by bites of infected ticks, infected flies, or infected fleas, or infected mites. So usually, because of this way of transmission, the portal of entry of this bacteria is the skin. And it is typical that Rickettsiae may spread via the bloodstream to infect the endothelial cells of the blood vessels and sometimes the vascular smooth muscle cells. The infection occurs due to the outer membrane proteins OMPA and OMPB. OMPA is the outer membrane proteins that are responsible for adhesion and entry into the host endothelial cells. So we blame outer membrane proteins in it. But then the basis of pathogenesis is how this particular species of Rickettsiae behaves itself, or genus behaves itself inside the human cell. That's really the understanding what kind of damage can be done by this particular type of Rickettsiae. So, usually, the Rickettsiae species enter their target cells by endocytosis or phagocytosis. So while they are internalized by these two possible ways, so the vacuole is rapidly produced, but then it will be degraded because the Rickettsiae may escape the phagocytic digestion. They may escape, as you can see here, in the cytosol. So this escape is mediated by one more very important virulence factor that is called phospholipase, produced by bacteria, and this phospholipase may dissolve the endosomal membrane. So then, once escaped from the endosome, Rickettsiae may freely multiply in the cytosol by binary fission. But then, of course, they cause the direct damage of the infected cells. And because it happens very often in the endothelial cells of blood vessels, the most typical sign of the very typical sign of the infection is vasculitis that is demonstrated as a rash on the surface of the body. So, and one more important stage in pathogenesis that Rickettsiae may move from cell to cell by actin mobilization, and we've seen those tail in the very first slide, and here somewhere you can see this is the okay, later on we discuss. So tissue biopsy, where when the cells are infected with Rickettsiae, reveals perivascular infiltration with lymphocytes, plasma cells, and polymorphonucleated cells, and histocytes with or without necrosis of the vessels. Gypsa staining is useful for an identification of the organism in the cytoplasm, and that's how we see it here. The vasculitis is most prominent sign of the infection, and it can be seen in the skin, heart, central nervous system, skeletal muscles, and kidney. That's why these make disease that severe. So here you can see the process of the attachment of Rickettsiae to the endothelial cells, and then they enter inside the cell via Rickettsiae-induced phagocytosis. And then, following phagocytosis, the phagosome membrane will be lost, and Rickettsiae enter directly the cytoplasm. So that what happens with the Rickettsiae in the infected cell. So there you can see that it starts dividing, as you usually see here, by binary fission, and it happens in cytosol. See it is indicated with the red or pink arrows. So Rickettsiae, then you can see the formation of the propeller, and by this propeller that is arranged in the human cell out of the skeleton components, cytoskeleton components. Rickettsiae will be propelled through the host cell cytoplasm, and the components of Rickettsiae stimulate the polymerization of the host cell F-actin. So the tail-like structure will be formed, and it helps Rickettsiae to travel within the cells. So, and here you can see that philopodia, and these are projections of the host cell of Rickettsiae, facilitate the spread of Rickettsiae to adjacent endothelial cells. So here you can see that how it had been proved. So various cells were infected with Rickettsiae connori and kept there for 3 days. Nucleic acid based assay had been staining had been done with a specific dye, and filamentous actin was labeled with fluorescein isothiocyanate, phalloidine, and rickettsial organisms were labeled with rabbit anti-rickettsial antibodies, so they are red. This is really indirect immunofluorescent. And then you can see the co-localization of Rickettsiae and filaments that are green with one pole of the organism in red, giving the appearance of the tail. So these are also connected with the organism. These are the tails formation for propelling the microbe between the cells. It is interesting that because it is intracellular bacteria, the most important protective immunity against Rickettsiae is mediated by the cell-mediated immunity. And interferon gamma and tumor necrosis factor mediated killing of Rickettsiae, and interferon gamma is produced by mainly T helper cells and to some extent NK cells, and tumor necrosis factor is produced mainly by macrophages. So these substances mediate the killing of Rickettsiae by facilitation of the creation of the autophagosomes. That is very typical for the cells infected with Rickettsiae. So you can see the so-called autophagosomes, that is the result of the autolysis and the digestion of dying bacteria. So then when Rickettsiae multiply in the bacterial cell, escape from phagosol and multiply in the cytosol. So then Rickettsiae or organisms related to Rickettsiae supposed to leave the cell. So, but then we have differences in the way how they may escape from the cell into the outside environment and cause new infection. So the most severe way of release from the infected cell is demonstrated by the Rickettsiae provazeki that causes lysis of the infected cell. My dears, this is the major event that is account for pathogenesis of the bacteria. They kill the cells where they multiply. While some other Rickettsiae-like organisms, they are not that tough. So, and for example, if we are talking about Rickettsiae, the causative agent of spotted fever, Rocky Mountain spotted fever group. So it first mediates the formation of the phylopodium and the focal lysis of the cell membrane, but don't cause the lysis, complete lysis of the cell, and that's how Rickettsiae act. And then the former Rickettsiae, now Arientsiae, that cause disease, recitin mice and coarse scrub typhus, Orientsiae tsutsugamushi. It is even softer. So, and it releases the cell from the cell by so-called budding, when the sort of bud is formed and bacteria escape from the infected cell without perforation of the endothelial cells. Few words more about the pathogenesis. And now we take a look at not Rickettsiae, but the second important or the third important group, that is Ehrlichia. So quite often the reservoir of Ehrlichia schaffensis, that is the most common one, is the deers, sheep, and dogs. So the disease caused by it is called human monocytic ehrlichiosis. And some other Ehrlichia, for example, Evingi and Anaplasma phagocytophilum. These are the cause of granulocytic ehrlichiosis, and rodents and horse can be the reservoir for it. One of the Rickettsiae-like microorganisms can cause so-called glandular fever in Japan. And that's also monocytic fever. It is a fluke-infected reservoir of this microbe is a fluke-infected fish. Both human monocytic and granulocytic ehrlichiosis is transmitted by tick bites. and within this bite the organism will be inoculated in the body and spread through the bloodstream. Macrophages or neutrophils may produce cytoplasmic vacuoles that contain many dividing Ehrlichia. Usually they divide like any other bacteria by binary fission. And you can see that the intracellular location protects them from being marked by antibodies and killed by antibody-dependent cytotoxicity. So here you can see Ehrlichia. And we have some transformation of Ehrlichia inside the host cell. So it will develop vacuoles, and there they occur in vacuoles as reticulate cells. You can see reticulate cells. Then as dense core cells will be produced, Ehrlichia containing vacuoles will be called morula. So then how Ehrlichia leave the cell? Ehrlichia usually infect leukocytes by phagocytosis, and then you can see the phagocytic vesicle. Then, as we underline, they multiply in phagosomes and multiply there, and they prevent the lysis of the lysosome or fusion of the lysosome with the phagosome, and while it is inhibited, Ehrlichia are not destroyed in the phagosome. So then when many bacteria divides already and increase in numbers, so we have the cell and phagosome lysis, so they cause just burst of the cells, that the cause of the granulocytic Ehrlichiosis, because mainly granulocytes will serve as a reservoir for the infection in a body. And you can see here Ehrlichia schafensis cell infection, and that's how it appears in infected cell. The last one that I want to discuss is Coxiella infection. So then maybe out of all Rickettsiae-like diseases, maybe it is most common, and it is typical zoonotic infection with a worldwide distribution, and the causative agent is called Coxiella burnetii. So usually several types of animals may serve as a reservoir, and usually these are domestic mammals like sheep, livestock, but also birds and arthropods may serve as a reservoir. So arthropods, because they survive, Coxiella survive for very long while inside the ticks' bodies. Interestingly, for this type of bacteria, the aerosol route is the primary mode of transmission. So, inhalation of infected fomites, or by the way, the injection of not pasteurized milk also can be the source of infection by Coxiella burnetii. So, contamination by Coxiella burnetii aerosols may occur directly from fluids of infected animals. For example, newborn animals from placenta fluid, or it may happen directly from the womb. So the organism may also be spread by the wind. So the Q fever may occur in patients without any evident contact with animals. So that's why it's so difficult to collect epidemiological anamnesis, because so that can be just transmitted as aerosol droplets. So that's why the lack of contact with animals may make us confused in the diagnosis of this disease. What is the pathogenesis of Coxiella infection? The thing is that out of all these Rickettsiae-like organisms, Coxiella may undergo phase variations related mainly to mutation in the lipopolysaccharide. So phase 1 is the natural phase found in infected animals, arthropods, or humans. It is highly infectious and corresponds to smooth lipopolysaccharide. The phase 2 organism is not very infectious and usually may be formed in the labs only after serial passages in cell cultures or embryonated egg cultures. And this type of disease is associated with the rough lipopolysaccharide. Compared to phase 1, phase 2 lipopolysaccharide displays a truncated LPS and lacks some proteins cell surface determinants. So how Coxiella leave the cells? Because, as I would like to repeat, the main event in in pathogenesis is how the Rickettsiae leave the cell, or Rickettsiae-like organisms. So Coxiella use specific eukaryotic receptor like integrins to invade the host cell. So the natural phase Coxiella burnetii usually poorly internalized by monocytes and macrophages, but also may survive within those cells. It is acidophilic bacteria. What does it mean? The metabolism is enhanced in lower pH. So regularly bacteria like neutral or slightly alkaline pH, and this one would like to grow under 4.7 to 5.2 pH. Therefore, it is very good adapted to the phagolysosomes. So if even the phagosome is fused with lysosome, Coxiella burnetii may multiply there. But if it is converted to phase 2 Coxiella burnetii, it is readily internalized, and Coxiellas of the second type may easily destroyed by macrophages in the phagolysosomal fusion. Usually Coxiella burnetii displays a complex intracellular cycle, leading to the formation of small metabolic infectious cells that are called spore-like forms. It is not sporogenic. Coxiella burnetii cannot form spores, but those structures that are produced that we call spore-like structure. So these are the forms that are released from the infected host cell, and it may be released by either lysis or possibly by exocytosis from the cytoplasm with Golgi apparatus. These extracellular spore-like forms gives this type of resistance to the environmental conditions that we have discussed at the very beginning. So, and it is resistant to desiccation or drying and low or high pH, and also It is resistant to chemical products like ammonium chloride and some other disinfectants and also UV radiation. That's why, because of the forming these spore-like structures, it may survive for a very long while outside the human body. And as we take a look at the pathogenesis, we see that it is readily ingested by phagocytes. So then the phagosomes are formed. Lysosome fusion occurs and bacteria may survive and multiply even if the acidification of the phagolysosome occur here. We mentioned that it's standing. And then they can be released from the phagocytes by lysis of phagosome and lysis of the whole cell. Few words about the diseases caused by these bacteria, by the Rickettsiae prowazekii, because this is the disease with very long history. And the first description of epidemic typhus comes out of the year 1083 from a convent near Salerno, Italy. But the most severe pandemics or epidemics of epidemic typhus usually accompany the major wars or some natural disasters. The biggest epidemic occurs during Napoleon's retreat from Moscow in 1812, when the more French soldiers died of typhus than from Russian bullets, it is believed. And it was said, or one could say, that lice have defeated Napoleon's armies. Moreover, their retreat has spread the infected lice and typhus through the entire continent of Europe when they come from Russia. So during and immediately after World War I, 30 million cases occurred with 3 million deaths out of epidemic typhus. It was really very serious disease. And if we take a look at the clinical findings, the clinical onset of epidemic typhus is usually abrupt. This is a very acute disease. And it occurs around 10 to 14 days after the infection. So that is the duration of the incubation period. So the first clinical manifestation we see is usually fever and very severe headache accompanied with abdominal pain. And fever is number one, but then other manifestation may include chills, rash. I would like to underline that rash is very typical also for typhus. So myalgia and arthralgia and central nerve system involvement with the delirium, coma and seizure development, because they spread and infect the endothelial cells, including endothelial cells of the brain blood vessels. Usually the disease is fatal in 10 to 30 cases. So then, it had been discovered that epidemic typhus may cause or may occur as recrudescence disease. This is a result of previous infection with Rickettsiae prowazekii. And then bacteria may perform persistency in the body for a very long while. And it was discovered in the United States. Currently, it occurs primarily in immigrants from the Eastern Europe who acquired epidemic typhus during World War II. And they had no history of the contact with the Laos in the new in the USA. So that's why they start thinking maybe that is the result of the activation of the microbe that had been already dormant in the body. And we believe that stress and waning of immune system and some other factors that influence immune status may help in reactivation of the Rickettsiae that reside in the body from the early infection. Usually the clinical symptoms of Rickettsiae disease are much milder than the primary epidemic typhus, because those who survive epidemic typhus, of course they develop antibodies, so they may have IgG or memory cells that may be activated if the hidden pathogen that occur in dormant state start multiplication again. Then the immune factors will be activated. So, how do we perform diagnosis of epidemic typhus and Rickettsiae disease? So, of course, microscopy is important. So, immunofluorescent and Giemsa staining can be used here. You can see the Giemsa staining. So, serology can be used and immunofluorescent detection of immunoglobulins M antibodies. So, we'll tell about the fresh acute infection. Usually it is performed with paired sera as we discussed for viral test. Some other tests like indirect hemagglutination assay and some other immunological tests had been used. I would like to draw your attention that culture method, it is extremely dangerous. So it cannot be easily performed. So that's why in the diagnosis, usually in a lot of laboratories, they try not to use the culture method. Usually immunological tests are used and also PCR tests now are developed. There is one historically important test that is called Weil-Felix test. And this test is based on the cross-reactivity of antigens of Proteus vulgaris. This is a bacteria from Rickettsiae. And these antigens of Proteus vulgaris may cross-react with anti-Rickettsial antibodies. So then it makes the diagnosis easier because it's very dangerous to work with Rickettsiae. So if we replace its antigen with Proteus vulgaris antigen, it makes this test much more available. But right now, I don't think it is used so with the development of ELISA and immunofluorescent test. That's rather historically important test, but is not in current use right now. So then culture reference labs only, not is used in routine diagnosis of the disease. So, treatment and prevention of epidemic typhus. So, because these bacteria don't develop that much resistance, the very old and still effective, still good antibiotics can be used for treatment. They are highly effective against epidemic typhus. But I would like to underline that tetracyclines are developed not for epidemic typhus. They are just the group of maybe most broad range of antibiotics, right? What does it mean broad range? So, because they may affect bacteria of many types. Any bacteria that may perform protein synthesis can be affected with tetracyclines. That's why even intracellular location of Rickettsiae will not protect them from tetracycline activity. Because antibiotic therapy does not eradicate rickettsia in lice-infested patients, because lysis or lysis may keep this rickettsia inside. The lousing is a very essential in the management of typhus outbreaks, because they may carry rickettsia bacilli for very long while. So if you are talking about control, specific prophylaxis of the epidemic typhus, whole phenol-killed vaccine is available. An attenuated live rickettsial Prowazeki vaccine against epidemic typhus now is developed and also available, but it is used not so frequently. But of course I have to underline that epidemic typhus disease now is extremely rare, and usually it may happen, for example, in the refugee camps, when a lot of people are kept together and we have the circulation of flies within the human population, only then, or if, for example, people are evacuated from the places of natural disaster and placed in really crowded conditions, then it may occur. But usually it doesn't occur in our normal life. So, I would like to say that first vaccine against epidemic typhus had been done by Rudolf Stefan Weigl during the World War II. And the production of this vaccine was based on the propagation of Rickettsiae prowazeki in the strain of clothes lice. So he used the vector to multiply Rickettsiae in this vector. So then the phenol whole-killed vaccine and attenuated live Rickettsiae prowazeki are available right now, but the first one that had been used in the culture in lice, now it's not really in use. So Rocky Mountain spotted fever. It had been described first as a specific clinical entity in 1899. And Howard Ricketts isolated the causative organism Rickettsiae in guinea pigs and demonstrated the circulation of this bacteria in between horse, mammals, and ticks in nature. And he also demonstrated that these rickettsia may transmit it through tick population transovarially. So it means all stages of ticks in particular area can be infected with this bacteria. So, but then this is endemic disease associated with the infection of the vectors in particular area. So the incubation period of this disease is shorter than that of the epidemic typhus, and it takes around 7 days since the tick bite. And the onset of the disease include high fever, headache, and malaise, and of course very dramatic red rash, and also accompanied with nausea, vomiting, anorexia, abdominal pain, and diarrhea. So here you can see the characteristic spotted rash. Usually it appears on the around 5th day of illness and heralds progression of the infection to more severe disease. So that's how Rocky Mountains spotted fever rash occurs, in which we see it closely. So it may result in various neurological manifestations, including deafness, convulsion, and hemiplegia. And That's because they most probably intensively grow in the endothelial cells of blood vessels. And of course, they interfere with the blood vessels' integrity and leakage of fluid and blood into vital organs such as brains. So that's why the neurological symptoms are common in this disease. So because edema will be produced, and in the perivascular space, and these asterics may result in the clinical encephalitis. So the disease is very severe. Так, we go ahead with the diagnosis. Microscopy. Rickettsiae will stain. In this slide it is said red, but I mean that's are there. So, or why I would say purple, right? So immunohistochemical detection of rickettsia is in a skin biopsy, a specimen of an eschar of the patient. The abundant inflammatory infiltrate is seen here with the necrotic features and vascular injury in the dermis. Diagnosis also can be performed by immunological tests, and all possible tests are developed. We may have limitations for it because several types of rickettsia may share the antigenic reactivity that we call cross-reactivity. And it always exists among antigens of pathogens within the same group, but even it may be shown in different groups and even in different genera. So right nowadays that PCR is used as a molecular tool, and this is a rapid diagnosis, and usually blood and skin biopsy can be used and subjected to PCR test. So the antibiotic treatment is very effective in controlling the infection. And apart from this, control of the vectors and reservoir hosts. Vaccines are developed against Rocky Mountain spotted fever that is made up of whole killed organism, but it doesn't give complete protection. And finally, Q fever causes flu-like illness at the beginning, and the incubation period may range from 1 to 3 weeks, and then very severe fatigue, fever, chills, and headache. It also may make us confuse in between just flu-like disease or flu disease and Q fever. So, but then usually the acute Q fever may have two very typical clinical presentations: atypical pneumonia and hepatitis. Then the chronic fever may be defined by clinical evolution lasting longer than 6 months. Endocarditis, the most frequent clinical consequences of chronic Q fever. And usually it is disseminated disease, but usually it's not fatal. So treatment is used, for treatment antibiotics are used, and human infections can be prevented by control of reservoir hosts, including vaccination of the possible hosts in animals. So for example, livestock can be vaccinated against Q fever. And vaccines usually contain attenuated organisms. With this, I would like to finish with Rickettsiae because I would like to tell at least few words and pass the tests for the one more topic for which we don't have a separate lecture and we don't have class for primates. But then you have to know at least something about them because they will be asked on examination. So what are prions? These are proteinaceous infectious particles. According to the protein-only hypothesis, what does it mean? Protein-only. It means that these particles don't contain DNA or RNA or nucleic acid. So they consist of protein only. And this is the only infectious agent that lack genetic material. So prion diseases are caused by the conformational conversion of the cellular prion protein. And the normal cellular prion protein is called PRPC. The C means cellular normal protein. And we believe that it can misfold into pathogenic form that is called PRPSC. So SC is the indication of the first ever found prion disease, and it was found in animals that is called scrapie. The initial letters of the word scrapie gives rise to this index, right? SC. Once formed, this prion PSC acts as a template to catalyze the conversion of other PRPC, normal PRPC in the body, into PSC, resulting in the accumulation of PRPSC, especially in the brains. Most cases of prion diseases in humans are sporadic, which means that PRPC spontaneously converts to PRPSC in vivo or is transmitted in an unidentified manner. So, prion proteins has been shown to participate in several biological processes. It means, what is the normal function of the PRPC? And because this is the result of the disease, right? So, but then because those PRPC proteins are mainly located in the brain, so then they participate in neuritogenesis, right? Neuronal homeostasis, cell signaling, and especially nerve system cell signaling, cell adhesion, and a protective role against stress. I would like to repeat, this is a normal function of the normal PRPC protein. Then misfolding involves the conformational conversion of the cellular protein to its misfolded pathogenic form, and you can see here how the percentage of alpha helix and beta sheets will be changing in normal protein, where B sheet makes around 3% of the structure, right? And this is a normal prion protein. And in pathological protein, B sheet makes around 40% of the structure. So, and this is a disease-causing protein where we have the prevalence of beta sheet structure. So that's why we call it misfolding. So, and here you can see that the prions are infectious protein that cause a variety of very rare but really invariably fatal neurodegenerative illnesses. And we have several types of illnesses in animals. For example, scrapie, that was the first one. But also we can have these type of diseases in humans. And in humans, of course, Creutzfeldt-Jacobs disease, chronic wasting disease, that's in deers, and bovine spongiform encephalopathy, that is called mad cow disease, but it may also occur in humans. So this agent can be transmitted to humans. And the disease itself is caused by accumulation of the misfolded proteins in the brains. You can see here that PRPC undergo conformational remodeling, the changes, and then PRPC is formed and then itself used as a template. It causes really refolding of the rest of the PRPC in the brains. So it means it may exist in two distinct conformational changes. But interestingly, these two forms, PRPC and PRPSC, may have different characteristics. So, ability to aggregate, PRPC don't have, and PRPC aggregate severely. Enriched beta sheet contact, no in PRPC and yes in pathological version. Interestingly, the pathological conformation of this protein is resistant to protease digestion. Most probably the site for proteolytic enzymes are protected by beta sheets. Then the pathological protein is neurotoxic, while the normal one is not. And the thing is also important that the pathological protein is infectious. It means if it enters the body by any of the possible route, for example, through GIT or directly as a result of hydrogenic procedure, when it can be transmitted from the brain of one patient to another one, but not correct disinfection of the brain chips, electrodes, right? This is for neurological transmission. Or it may be transmitted through retina because it may be accumulated also there, not only in brain zones only. So it may cause infection. But the thing is that infection caused by PRPSC, it is not really, it doesn't look like regular infection. Why? Because there is no inflammatory response within the spreading of this pathogen. So there will be no temperature and there will be no other signs of inflammation, and there will be no immune response in the body. That makes the diagnosis of prion disease very difficult, but also it's because they have a very long incubation period. It is believed that Creutzfeldt-Jakob disease has an incubation period around 30 years. So here you can see a very nice scanning microphotograph where it is shown like prion proteins that are shown in red can become infectious and cause neurodegenerative diseases, and you can see 4 nerve mouse cells illustrating how infectious prion proteins may move within the cells, right? And they move along the neurites, wire-like connections the nerve cells use for communicating with adjacent cells. And here you can see the accumulation of the prion proteins. So here we can see the brief, in one single slide, we can see the brief history of prion study. So it is interestingly that the first disease caused by prion had been identified in sheep in 1732, and it had been called scrapie. And first it had been described. And scrapie is referred to the sound of poor animals, that their skin is itchy. and they lose the contact sensitivity, and they rub their body against fences until bleeding, because they don't feel pain, but they feel itching. So then scrape is the sound of this rubbing. So that's how they call it as a disease. And then you can see the disease had been discovered before the viruses had been discovered, right? So you can see here Ivanovsky works and Bejer-Link, who identified viruses, but then it had been seen later on that this is not viral disease. And here you can see in 1920, the first case of Creutzfeldt-Jakob disease had been described. And then I would like to draw your attention to the 1954. Then Sigurdsson proposes scrapie caused by slow virus. And first cases of Kuru human disease had been discovered in that time. And then in 1959, Gaidusha traveled to the Papua New Guinea and he studied the Kuru disease. And he did a lot to explain the epidemiology of the disease. I would like to show you a couple of slides. So, Kuru is a rare and fatal nervous system disease, but it appeared that it is epidemic, and it reached the epidemic sites in New Guinea for people. And Gaidusha demonstrated that for people, aborigines of the New Guinea, contracted this disease by performing ritual cannibalism of corpses during funeral rituals. So, and the name Kuru means to shiver or trembling in fears. So, because those people, and you can see here the Papua New Guinea Aborigines, symptoms of the disease included muscle twitching and loss of coordination. And you can see here the development of the, here we can see least severe symptoms, then they progress to the more severe symptoms, including difficulty with walking, involuntary movements, behavioral and mood changes, dementia, difficulties with eating and swallowing food, right? And then, without any doubts, death will occur. So, and that was the Gaidusha who discovered the spreading of this disease through ritual cannibalism. And by the way, he was given the Nobel Prize for it. And I also would like to underline the works of Stanley Prusiner in 1982, who also was awarded with the Nobel Prize, who demonstrated stability of the scrapie agent, and he developed the name prion for proteinaceous infections. And he also demonstrated the so-called transmissibility of this disease, because he took the brains from the infected post-mortem samples, right? And he injected mice with these post-mortem samples. And within half a year, mice develop so-called spongiform encephalopathy. All those diseases are used to cause so-called spongiform encephalopathies. What we refer to spongiform as? This is the appearance of the brain tissues. You can see here the brain section showing spongiform pathology characteristic to all prion diseases. And to develop briefly, the pathogenesis occurs within these stages. misfolding of the normal protein into PRSC protein, then formation of amyloid prions and rods, and then amyloid plaques, and then accumulation of them and the development of spongiform degeneration. My dears, pay attention that this way takes decades, but then only after spongiform degeneration occurs, the clinical symptoms will appear. So it means that the brains are damaged already only then we may know by clinical symptoms that disease occurred, because before it is asymptomatic. So out of this pathogenesis, we can see the potential target stages for treatment. But I would like to tell you the treatment is not really developed yet. But the potential stages are reduction of normal expression of PRPC, right? So, but it may influence the normal functioning of the brain, so that's why it's not really very good. Then, most probably, we have to employ the approach of inhibiting conversion of PRPC into PRPSC. And also we may stimulate the clearance of PRPCs from the body. And also we may somehow block the neurotoxicity of already developed misfolded PRPC. So, and that is important because if we are talking about the diagnosis of spongiform, they are called altogether transmissible spongiform encephalopathies. So you have to get some information about that. First, pre-mortem diagnostic tests are not available. See, no pre-mortem diagnostic tests. What does it mean? It means if we take a look at pathogenesis, if clinical symptoms appear, the patient gonna die anyway. So by now, we cannot really, the tests are not developed to perform diagnosis before the clinical symptoms appear. So usually we perform diagnosis based on clinical symptoms if we are talking about pre-mortem diagnosis. Then the definitive diagnosis is post-mortem through brain histological analysis showing spongiform changes, astrogliosis, and sometimes by showing the amyloid plaques that occur in Kuru like this and in variants of Rickettsiae disease caused by transmissible prions like this. So, but this is post-mortem sample, post-mortem diagnosis. Then, as I said, clinical signs are used to diagnose human TSE diseases. And pre-symptomatic diagnosis is currently impossible. So, on the stage of pathogenesis, we don't know about it, we cannot diagnose this disease. So, but the good news are that new strategies are being adopted right now to try to develop pre-symptomatic diagnostics. We believe that before we were absolutely sure that no immune response is produced, right? But so-called conformational antibodies can be developed against not the normal PRPC, but against pathological prion protein because it is enriched with beta sheets. Then some spectroscopic techniques and amplification of altered prion isoforms. Those techniques are in trials, not in human trials. So we have just animals where they try to introduce these new pre-symptomatic and pre-mortem diagnostic techniques. With this, I would like to finish. I see that we have over time already. And again, so my dears, so these are the topics that can be seen in the exam question. See you next time, my dears, and we finish with this lecture bacteriology. The next lecture will be in special virology. Goodbye, my dears. See you in the classes.